Assalamu alaikum. I am a student of BS Chemistry. The topic that I am going to present is a Baeyer-Villiger oxidation. So first of all, what is Baeyer-Villiger oxidation? Basically, it is an organic reaction that involves the conversion of ketone into an esters by using oxidizing agents that are per acids or peroxy acids like metachloroperidoxybenzoic acid, hydrogen peroxides, these kinds of per acid or peroxy acids. But if we're using cyclic ketones as a substrate, then we will convert into a cyclic esters, and the cyclic esters are also known as lactones. And if we treat aldehydes with same peroxides or peroxy acids, then they will convert it into carboxylic acid, corresponding carboxylic acid. But in some cases, they will also convert into a formate ester. It will depend upon the relative migratory aptitude of alkyl group with respect to hydrogen. Next, we will discuss about its historical background. In 1899, there were two scientists, Adolf Baeyer and Victor Villiger, that first gave demonstration of this oxidation. They used peroxy monosulfuric acid, an oxidizing agent, to convert the cyclic ketones into an cyclic esters, means lactones. And with cyclic ketones, they used camphor, menthone, and tetrahydrocarbon. All these are cyclic ketones. And from these two scientists, the one Baeyer was more famous, and he was awarded the Nobel Prize in chemistry in 1905. Next, there are some general reactions of this Baeyer-Villiger oxidation. We can see that ketones are converted into corresponding esters by using same per acid or peroxy acid as an oxidizing agent. While in case of aldehydes, when they are treated with per acid or peroxy acids, they are converted here into carboxylic acid or formate ester, that is shown here. Next, we will discuss about its mechanism. It includes first dissociation step, in which per acid or peroxy acid that we use as an oxidizing agent, it will dissociate to give proton. And in second step, that is a protonation, in which the carbonyl compound, like we are using ketone, it will protonate it by using one of the lone pair of oxygen. Now this protonated oxygen that will carry positive charge, it will become more electron withdrawing, so it withdraws electron density from the adjacent carbonyl carbon. As a result, carbonyl carbon becomes more electrophilic, due to which the conjugate base that is produced in the first dissociation step, in which per acid get dissociated to give proton and a conjugate base. This conjugate base will act as a nucleophile and attack on the electrophilic carbon of the carbonyl group. As a result of this attack of nucleophile on this carbonyl carbon that acts as an electrophile, we will get a tetrahedral intermediate. Next step is the product formation step. As in case of intermediate, when a carbon attached with the two electronegative atoms, it becomes more unstable, as here carbon is attached with the two oxygen atoms. So it will collapse and bond breakage take place, as here. So here, from one of the lone pair of the oxygen atom that is present on the top, it will move inward as a red circled oxygen. One of its lone pair will move inward to form a double bond, and the first arrow indicates that this alkyl group will migrate towards the next oxygen atom. As a result of this migration towards the next oxygen atom, the next arrow that is present between the two oxygen atom is here. It is showing that this bond will break. As a result, we will obtain our product, that is the ester in the protonated form, and conjugate base will abstract the proton and convert into the corresponding ester. So this was all about its mechanism, and next we will discuss about its synthetic applications. Synthetic applications of this Baeyer-Villiger oxidation. So here first one is the synthesis of testololactone, in which dehydroepiandrosterone are converted into testolactone in the presence of Baeyer-Villiger monooxygenase. These are basically enzymes that catalyze this Baeyer-Villiger oxidation. So through the mechanism of Baeyer-Villiger oxidation, we can synthesize testolactone. Next we will discuss about its mechanism. As we have already discussed about its general mechanism of this Baeyer-Villiger oxidation, so same, first is a dissociation step, in which the per acid that we are using in this step, testolactone synthesis, will get dissociated to give proton. And the second step, the carbonyl carbon will get protonated. Next step is the formation of intermediate. As we have already discussed that when the oxygen gets a positive charge, it will become more electron withdrawing. So withdraw the electron density, form the adjacent carbonyl carbon, and this carbonyl will get more electrophilic, and the conjugate base that will produce in the first step, it will act as a nucleophile here and will attack on the carbonyl carbon. And when it will attack on the carbonyl carbon, one of its bond will be shifted towards the oxygen atom to stabilize its positive charge, and we will get a tetrahedral intermediate. And next, one of the alkyl group will migrate. As a tertiary alkyl group has a greater migratory aptitude, so it will migrate and a bond between two oxygen atom will break. Here red arrow is showing that from here the migration towards the oxygen atom take place, and the black arrow will indicate that how this group will migrate towards the oxygen, and the bond that is present between the two oxygen atoms will break, and we will get a protonated carbonyl compound and anion in which oxygen carry a negative charge. This will abstract the proton from this protonated carbonyl compound and will convert into the corresponding ester. And here you will notice that here is a cyclic ester is produced, so it is known as a lactone and its name is a testolactone. Next we'll see applications of this testolactone. The conversion of the dehydroepiandrosterone to a testolactone by the use of this Baeyer-Villiger oxidation was first reported in 2013 by Alina Switzer. And it was also reported that this Baeyer-Villiger oxidation was catalyzed by Baeyer-Villiger monooxygenase, that are enzymes basically microbial that are produced by fungi. And this testolactone has an anti-cancer, act as an anti-cancer agent, that is also known as an anti-neoplastic agent. Anti-neoplastic agent, basically those are medications that are used to treat cancer. So it is used to treat breast cancer. Next is the synthesis of Taxol. It is also one of its application of a Baeyer-Villiger oxidation. Basically, it is completed in the 21 steps. It also includes a Baeyer-Villiger oxidation step. So first, in the laboratory of Tony K.M. Cheng, this Taxol was synthesized from carbon. One of its enantiomer, that has the S configuration of carbon. Carbon basically cyclic monoterpene ketone that belongs to a terpenoids chemical family. Here, some key steps are mentioned in the synthesis of Taxol. There is a Baeyer-Villiger oxidation, as we have discussed here. Oppenheimer oxidation, that it is a basically oxidation of secondary alcohols to ketones. And Meerwein-Pendorf-Barley reduction, it is a reduction of aldehydes and ketones to their corresponding alcohols in the presence of aluminium alkoxide as a catalyst, and alcohols like methanol, ethanol or glycerol can be used. And Grignard addition, intramolecular nucleophilic substitution reaction. In the given reaction, it has shown that ketone is converted to its corresponding ester, and after several steps, it is converted into the Taxol, that is its final product. Now we will discuss about its mechanism. Its mechanism is also first dissociation step, in which the metachloroperoxybenzoic acid gets dissociated into its conjugate base and a proton. And the second step is the protonation step, in which the protonation of the carbonyl group takes place. And next step is the formation of intermediate, in which a conjugate base as a nucleophile here attacks on the carbonyl carbon, and one of the double bond is shifted on the oxygen atom. Here, by the attack of a nucleophile on the carbonyl carbon, the formation of tetrahedral intermediate is shown. As in this intermediate, the carbon is attached to the two electronegative oxygen atoms, so migration of alkyl groups towards the oxygen and will take place, as here it is shown by red colored arrow, that this alkyl group migrates. And then the bond between the two oxygen atoms will break, that results in the formation of protonated ester and the carboxylate ion. This carboxylate ion will abstract the proton from this protonated ester and result the formation of the ester plus carboxylic acid. Next are the applications of Taxol. Taxol has also anti-cancer properties. It is used with some other drugs that are anti-cancer like adriamycin, vinorelbine, cytoxin, cisplatinum. These are all the anti-cancer drugs. So Taxol is used along with drugs to treat several types of cancer like ovarian cancer, head cancer, neck, breast, lung cancer. Another example is the formation of benzoic acid from benzaldehyde by Baeyer-Villiger oxidation. As we have discussed that by treating aldehydes with the Baeyer-Villiger monooxygenase enzymes, upper acid, upper oxy acids, these are converted into corresponding carboxylic acids. Because here on one side phenyl ring and on the other side hydrogen is present, and here hydride due to its smaller size it has a greater migratory aptitude, so it will migrate and corresponding carboxylic acid will be produced rather than ester in case of aldehyde. But in some cases, if on the phenyl ring electron donating groups are present like methoxy or alkyl groups are present, then in this case they have a greater migratory aptitude. So in this case formate ester will be formed. Next are the applications of this benzoic acid. Benzoic acid basically used in the industry to prepare several kind of products like perfumes, dyes, topical drugs. Topical means these are drugs that are antiseptic like antiviral, antifungal that are applied on skin are insect repellents. Its sodium benzoate is also used to preserve the food to prevent its from microbes because it has an antimicrobial characteristics. And acid it is also used as an inactive ingredient in the synthesis of various medicines. Inactive ingredient they are as a flavoring or to stabilize the various kinds of medicines and to increase their absorption in the body, and it can also be used as a lubricant in capsules or tablets in order to prevent their fragmentation. So this was all about Baeyer-Villiger oxidation, its mechanism and its synthetic applications. Thanks for watching.